Good.
Great. Go ahead, Anmar.
Okay, 1 second. I had the window open, but I closed it. Okay, anyhow, now it's open. So welcome everyone to today's QLS, Kanban, Ludmer Center, etc. talk. Our today's speaker is Ben Goult. Ben comes from University of Kent. So let me give you a brief history about his academic career so far. So he basically finished his undergrad degree in 1998 from the University of Sheffield in biochemistry. Then he went on to do his PhD at University of Manchester Institute of Science and Technology in biological science in 2002. Then he went on to become a research associate again at the University of Manchester between 2003 and 2005. Then he went to the industry for some time. He worked for AstraZeneca. So I don't know if he participated in producing the vaccine, but I guess that was a while ago, so maybe not. And he stayed there until 2006, then returned back to academia as a research associate at the University of Leicester, and became a research fellow between 2006 until 2014. Then finally, he joined the University of Kent as a lecturer in 2014 and stayed there ever since, going through the ranks from lecturer to senior lecturer, and now he's a reader, which basically is equivalent to being a full professor here in North America. His research interest is mostly in structural mechanobiology that combines structural biology, biochemistry, biophysics, and mechanobiology to define the role of how physical and mechanical forces are sensed through the cell extracellular matrix and adhesion complexes to control cellular processes. He also developed this theory related to Talin, which basically try to define define it as a signaling hub and as a mechanical protein that allows ion channels in defining their electrical properties, and that's pretty much the main probably topic of this talk today. So thanks, Finn, for joining us, and we look forward to hearing your talk. So go ahead.
Brilliant, thanks, Emma, for the nice detailed introduction, and thanks to Celia and Alex for the invitation and organising this. It's great to present. It's just a shame I couldn't actually come and present to you over there, although it sounds a bit cold for me. So yeah, as Emma pointed out, I've been working, well, actually, I don't know if you did point out, but I've been working on this molecule Talin since 2006. And when I was in industry at AstraZeneca, I was commissioned to test some new software. And one of the new bits of software was done by a guy at Leicester who was working on Talin. So the model data he sent me to work on was a chunk of Talin. I don't know if I've got my cursor. Do you see my cursor, actually?
Not right now.
So I was actually just working on this little bit of Talin just as a test case. And from that, it led to discussions, and then ended up leaving industry to move to Leicester to work with David Critchley to work on this molecule Talin. And I've been working on it ever since. We're trying to understand what it does. And the more we find out, the more interesting this molecule gets. And what I'd like to present to you today is a new idea which emerged from the experiments we've been doing on that this molecule has the capacity to store information, binary information, into the shapes of the molecules. So I'd like to hopefully convince you that Talins are memory molecules, and which leads to this rather provocative title of Does Our Brain Store Memories in a Binary Format? And hopefully over the next 40 minutes or so, we can have lead to a discussion on that. Oh, I can't change the slide. Okay, so in terms of how the brain works, and we all know it from like primary school kids right up to everybody knows about how the brain works. We've got this soft lump in our brain, in our heads, And it's comprised of trillions of synapses and trillions of neurons all talking to each other in a highly ordered way, sending electrical and chemical signals around across these linkages between cells. These are cell-cell junction connection where they can talk to each other, these synapses. And all of this electrical and chemical signaling looping around gives rise to complex calculations and complex running of a complex organism. And this is all pretty well mapped out and it's generally known, but what's not so well known, oh dear, is how the information is actually stored. You've got this electrical and chemical signaling coordinating the organism, but how's it physically captured and updated and worked upon? And what I'd like to lead up to is this notion based on this discovery of these binary switches which are built into each of these synapses. So these synapses are where two neurons are making a contact and one can send a message to the other one and then it can send a message to another and they can all update each other. In these cartoons, these pretty cartoons, you don't actually see that these two nodes of the synapse are held together by a complex scaffolding which holds them at this perfect distance apart to allow electrical chemical signaling to occur with a meticulous precision. And that scaffolding is essential. If you knock out any of the scaffolding partners, then these synapses don't assemble properly. And in this model, which I'm presenting here, this scaffolding is built of these memory molecules with these binary switches in them. So what I'd like to try and argue today is that these memory molecules built into each of the synapses in the brain, maybe their memory molecules are used for memory in the context of a more complex organism. And that the synaptic scaffolds represent this complex array of binary switches. And this led to this paper which came out earlier in the year on a new theory. It's basically the culmination of the last 15 years of my research onto Talin to present an idea of what might be going on if these systems were ordered and if there was more complexity to act than what we currently imagine. So if you're interested, you can Google MeshCODE and this is the top hit. But before I can explain that, what I'd really like to do is just to go back quite a few steps to talk about our day-to-day research and our interests and how it leads up to this concept. And we focus, as Amma pointed out, we focus on the adhesions that all animal cells make to this underlying protein meshwork called the extracellular matrix. And these cells physically grab onto this matrix. And on this video here, you can, on this photo here, you can see where this cell is physically attached onto that cargo-like matrix by these yellow adhesions. These are large multi-molecule, multi-protein complexes, and they're essential for, I would argue, all cellular processes, which is arguable, but that's my position. And these attachments are mediated via these integrin family of cell adhesion molecules. And I'm not going to go through all of these in detail, but one I would just like to flag at the start is this essential role and the regulation of gene expression and how cells can differentiate and specialize as a function of these attachment points. And this is best shown by an experiment from Adam Engler and Dennis Disher. It's over 15 years old now, but it's an absolutely beautiful experiment in my opinion. And it's to take a population of these mesenchymal stem cells. And these guys they're not differentiated yet. They've got the capacity to reprogram into many different lineages. And what the experiment here is beautifully simple because normally this cell will receive different signals, both chemical, electrical, hormonal, all of these different things and mechanical, and then it will use those to remodel. But in this experiment, it's the only variable in this experiment is the stiffness of the matrix that this cell is sitting on. So these little blue squares where it grabs onto that matrix are attaching onto it and they just vary this stiffness. And what you can see is if you put a stem cell onto something like jelly, it will reprogram and turn on a different cellular program to become a fat cell or a brain cell. If you put it onto a glass-like substance, it will remodel and turn into a bone cell. So clearly what we've got here is we've got a complicated computational step which is occurring in when these cells physically grab on and interrogate the surface that they're on. And what this tells us is that adhesions are information processing centers. And that's what we want to understand. How can these little tiny blue squares do this and totally lead to global reprogramming of the whole cell. So if we go into one of those blue squares in a little bit more detail, then what we see is we see large multimolecular complexes. And here we've got these red integrins. These are the hands of the cell. These are what physically grab onto the extracellular matrix, which is shown here in blue. So there's hundreds of these hands which all grab on and hold the cell in the right place. And then you've got the plasma membrane just here. And just inside the cell on the plasma membrane, you've got this dense cluster of talin and vinculin molecules in what's known as the integrin signaling layer. And then these talins and vinculins directly attach these integrins to the cell's cytoskeleton. And the cytoskeleton is made of actin and microtubules, but it's the fourth generation machinery. The cell's got motor proteins which can pull on these blue fibers and generate forces. And these forces can push and pull onto these linkages. And this is the focus of our research and the focus of today's talk, this computational layer here. And if we go in a little bit more detail to now look at the individual proteins, then what we can see, and this has been flipped 180 degrees, so now the matrix is at the top, but what we're showing here is a single integrin, one of these hands of the cell, grabbing onto the matrix here. And the integrins can switch between a fist closing up and grabbing on, and that enables the cell to grab on, let go, grab on in response to signals. And one of the major signals which drives that process is the protein talin, which can grab these small cytoplasmic tails of the integrins and stabilize this adherent state. So when talin grabs on, the integrin will grab onto the matrix. And then importantly, what talin also does is it directly couples the matrix integrin complexes to the cell's fourth generation machinery. And this is shown here by these yellow circles. This is actin, and this can generate forces to pull on these complexes. And what this means is that talin's in a fantastic spot because it can detect forces from outside the cell if there's movement, or inside if the cell switches on its motors, and it can respond to those forces. And the classical view of talin's mechanosensing is that when you apply force to talin, you recruit a protein called vinculin, and vinculin cross-links talin to actins and makes these adhesions big, makes them strong, and makes them grow. What I'd like to flag here is that what I'm defining here is three proteins, integrin, talin, and actin. And these form the core of all of these, not all, but nearly all of these adhesive complexes. But what's really striking there is that the structures which you can assemble on an integrin, talin, actin core are widely different, and depending on the mechanical properties, depending on what other proteins are in there. And they go from this beautiful video here from Mark Morgan in Liverpool, showing a fibroblast crawling along a matrix. These adhesions in green, they get big at the front, they contract and pull against those adhesions, then they disassemble at the back. Really dynamic, really tightly regulated to enable our cells to crawl to where they need to go. Just on the west coast from you guys in Vancouver, Guy Tannensath's lab looking at fly embryos. Here we've got a Drosophila just at the first moment where it becomes contractile, and the muscles shown in red, and this yellow layer, this yellowy green layer, that's the integrin, talin, actin holding the muscles together. Over on the right, we've got a cell which is dividing, and the cell has to be able to go from that pancake shape at the start. It's really hard to cut a pancake in half. So what happens is the cell rounds up, it lets go of its adhesions, rounds up, and then once you're in a sphere, it's easy to have poles and go in 2 dimensions, and then you immediately spread. But that's absolutely dependent on the integrin, talin, actin linkages to let go and grab on at the right time. And these I won't talk about, but what I want to focus on in the final part of my talk today is these adhesions which hold all of our synapses together. Again, this is ECM, integrins, talin, and actin, and it's essential for this. If you knock out integrins or ECM components or contractility, these things just disassemble. So clearly there's a lot of different things that you can do with the talin molecule. And when I moved to Dave Critchley's lab in 2006, we were working on what this molecule looked like. Back then we were just interested in how it was this fundamental adapter. And this slide here summarizes about 5 years work between 4 postdocs, me, Neil Bate, Alex Gingrass, and Igor Barsukov, who was just going independent. But working with Dave, we worked hard to work out what this looked like, and we identified that Talin looks like this. And I don't want to go into a lot of detail here, just to point out, it's got an N-terminal region which binds to integrin, and in the simplest linkage, we've got a C-terminal actin binding site at the end. But between those two attachment points, between the attachment to the surface and the motors, we've got 13 of these helical bundles, which once we knew there was 13, we renamed them to R1 to R13. Anyone familiar with the Talin literature before that, it was a total mess, but now these are the 13 switches, R1 to R13. And once you generate force, this is all in the force transmission pathway. And why these are switches is that they have a special property that they have, they can adopt two thermodynamically stable states. In the absence of force, or at low force, they stay in this folded conformation, but force can open them up into this conformation on the right. And then if you lower the force, they don't refold straight away because there's hysteresis, but if the force goes away, then they'll reset back to this state. So what we've got is you've got this, each of these switch domains has got two thermodynamically stable states, and they can open and close between these states using mechanical forces. And what we now want to do to clarify this and to make the nomenclature easier is we now call this folded, this closed O state, and this open 1 state. And of course, when we were doing the structures of this, we didn't just think, oh, these look like switches. The reason we had this sort of epiphany that these were switches came from some biochemical analysis, which we were doing in the lab, where we were looking at a ligand of Talin, which is an effector for the small GTPase, RAP1. And this guy is called RIAM, or RIAM over your side. But RIAM binds to this switch, and it binds via what's becoming a really common mechanism in this game, is it binds by a mechanism called helix adhesion, where the helix of RIAM packs onto the side of the helix of the bundle of Talin, such that when this domain unfolds, the binding site's gone, so that ligand goes away. But what happens simultaneously is you expose this vinculin binding site in red. So immediately we could see that this is a binary switch. We can switch between two different signaling processes, two different jobs that the cell's doing, by causing the transition between this closed O state and this open 1 state. And I think this is almost like a textbook example now of how you could sense forces and convert them into biological signals. And the obvious question, this was just as I was setting up my own lab back in 2014, the obvious question was, how much force do you need to pull on a molecule to cause these switches to open up, to cause this change in behavior? And it was that question which led me to contact Ji Yan in Singapore. And I would say just a massive shout out here to Ji Yan and Ming Chi Yao, because this is, and Mike, but particularly to Ji, this has been the best collaboration, and every single experiment just is mind-blowing, and we've learned so much, and they're good friends as well, so a big acknowledgement to these guys. But the experiment which we first set out to do was to just know how much force do you need to apply to make this R3 domain switch open. So the experiment is conceptually very simple. You can take a single molecule, just one molecule, covalently engineer it so you can covalently attach it to the cover slip of a microscope via its N-terminus, and then via its C-terminus, we can attach it to a magnetic bead. And what this enables us to do is with a small permanent magnet, we can pull on the bead and move it away and exert forces onto this molecule, which enables us to see how that molecule behaves and how it interacts under force. And I'm not going to show a lot of data from this paper because it's a few years old now, but just to highlight what this data looks like. So in this experiment, we're pulling on the bead, so we're increasing the force onto the molecule going along the x-axis, and then we're measuring the extension, the distance of the bead from the surface. So as we pull on this molecule, it starts to get pulled taut, not surprisingly, but what you start to then see is these jumps in the extension, and that's where each of these domains, these 3 domains, 1, 2, and 3, unfold independently. So these are these domains behaving as completely discrete switches opening as a function of force. And each of them's got its own force threshold, the amount of force you need to make it open. And again, not to go into detail here, but one of the things we've been very keen to do is to engineer these switches to make them more or less stable, to actually reprogram this thing in essence. But we can stabilize R3, and we can show that that initial unfolding step here is R3, because if we stabilize it, we can make it need a much higher force to open. So to summarize our first understanding of one of these switches, this R3 represents a key checkpoint in adhesion assembly. And it's really an important checkpoint because these are massive complexes which are going to form. You don't want them to just start to form in the wrong place at the wrong time. So it's worth just spending 30 seconds to go through this. So here we've got a cell which is migrating down towards the bottom right of my screen. And right at the front of this cell, you get a lot of RAP1 signaling. So you start to recruit a lot of this RAP1 effector REAM. And REAM binds to that folded R3 domain. And in doing so, it brings Talin to the front of the cell. And all of the components start to assemble and come together at the front of the cell, ready to start to unpack and make these adhesions. And once their Talin can open up, it can begin to grab onto the integrins and it can start to activate them. If you think about this in terms of forces on Talin, they're negligible because even if it grabs on at this end, without something pulling on the other end, it's a low force condition. However, you can get to a key checkpoint where a Talin activates an integrin, which grabs on and simultaneously attaches to an actin filament. Now, all of a sudden, this molecule's under tension and the forces can start to build up on that molecule. And the first thing to happen is that the R3 domain unfolds. You bring vinculin in, which then reinforces these adhesions, and then they can grow bigger and bigger until you end up with these large things called focal adhesions, which are very vinculin rich, very big. But the R3 is the checkpoint to make sure that these complexes only happen once you've made a successful tether. So having identified a key role for this R3, we were interested to know, have all of these switches got the same capacity for this switch-like behavior? Not least because in the meantime, we've got over 40 different ligands which are decorating this thing. So we're interested to know, is this whole molecule as mechanically sensitive? And this figure, it's published a few years ago now, but it still makes my jaw drop every time I see it, because here what we're looking at is we're stretching one Talin molecule of the whole rod, R1 to R13. So these 13 switches. And when we start to increase the force, exactly the same as before, at about 5 piconewtons, R3 unfolds. That's that adhesion triggering and starting to build. But then what we see this staircase-like behavior. Each of these steps is one of these domains unfolding independently. So they don't unfold in like pulling a thread on a jumper where the whole thing goes. They go one and then another, because as they unfold, they actually introduce a drop in tension because it suddenly gets stretched. But anyway, these steps, each of them represents one domain unfolding. And because each domain binds a protein in its folded and unfolded state, then each of these steps represents a domain mechanosensing and changing the biological signaling in response to force. I should also just flag here that this is 6 cycles. So you pull it till it's completely spaghetti and then you let go. And then it immediately resets itself if there's no other components in there. So this is what you'd want with a mechanosensor. If there's no force, then you go back to your default state. Where this gets interesting is if you start to look at the molecules which are decorating the switches, and this is a really abridged list because it's expanding all the time. And as we do proteomics in different tissues, we find different ligands. So this is a constantly changing set. But if you look at the different molecules on this list, they pretty much define all walks of life of the cell. You've got transcription factors, you've got gaps and GEFs, which are remodeling the cytoskeleton. You've got kinases, you've got cell cycle regulators like the CDK1, you've got microtubule regulators, etc. And they're all binding onto these switches as a function of the switch pattern that they're in. So you're basically turning on and off different cellular processes. And one of the things which we're interested to do, and I'm not going to go through them all, so don't panic, is to say, what do each of these switches actually do? What's their cellular roles? And as I've already mentioned, the R3 is what switches on adhesion. I just want to flag 2 here, just because I think they're pertinent. So the R7 domain, what we've found is that it directly determines whether microtubules target to these adhesion sites. And because when it's in this folded O state, it's got a high affinity binding site for a protein called CANK, which is part of a microtubule stabilizing complex. So when this is folded, microtubules target to adhesions, When this is open, microtubules don't target to adhesions. And then also this one here, R8, and this is a paper which we published earlier in the year, this directly couples adhesion to the cell cycle, because the CDK1, the cyclin-dependent kinases, sit on this domain and they phosphorylate these switches and change the order which they unfold. So you got global reprogramming of the mechanotransduction during the cell cycle. But what this means is we've got a really complex problem to work on, because we've got a protein, and so far I've only talked about one molecule, but we've got a molecule which has got 13 switches. It's getting pushed and pulled in lots of different directions by different force generating machinery with different trajectories and different angles. And so what we start to imagine is that we've got this complex coding, this Talin code, where this protein is constantly decorated with signaling molecules. And if you convert it into this binary format, you could almost, this is a 0000 state, but it's got one set of ligands bound to it. But as we increase force and we cause one of these transitions, we change the signaling on that adhesion, on that Talin, by recruiting one player and displacing another. So this guy is free to now diffuse off into the nucleus or go wherever he wants to go. But you're changing the binary information in that molecule, and you're changing the readout because the signals are different. And then you pull again and you further change this. So even on a single molecule in an adhesion, there's a complex coding which is going on. And for those who are interested, there's a poster which came out in this month's Journal of Cell Science where we tried to summarize this in a poster form. And some of the images I've got in today's talk are actually lifted straight from this. But what leads to it is the hypothesis that Talin is directly regulating a wide range of cellular functions through these switches opening and closing to modulate these binding interactions. And that's where we got this notion of this mechanosensitive signaling hub, because you get different platforms depending on the mechanical environment and what factors and what proteins are available. But if you take this into, just consider that there's many, many different competitive interactions for these sites, and there's 13 switches, and then there's many, many different force vectors, and it leads to this idea that there must be a code that underpins all this to enable this robust, reproducible signaling responses, which remain metastable in response to diverse multiple inputs, because we form from a single fertilized egg with really high fidelity. And this whole thing works out, this mechanical computation up against the genomic coding. It gives rise to a highly ordered being. And that's what we want to decipher. We want to decipher this mechanotransduction code running through Talin. And where this gets complicated is, so far I've only talked about a single Talin molecule. If you look at what these adhesive structures look like, you've actually got a complex meshwork of molecules, lots and lots of Talins. Also, I've not shown it, but vinculin itself has got 6 switches. So just with Talin and vinculin, there's like a vast number of switch patterns you can arrange. And they're not the only mechanosensitive proteins in these sites. So this is a really stripped down version. But what happens then is, depending on the pattern of 1s and 0s, you recruit all of your favorite signaling molecules onto that site as a function of the patterns which exist, and in a context-dependent manner. So what we start to imagine is on the bottom of our cells in tissues and around the edge of the synapses in the brain maybe, but in the bottom of our cells, we've got this constant mechanosignaling meshwork, which is reading and interrogating, constantly responding to the physical environment. And it's able to sense and respond because of these switches. But just to clarify this, if you ask a couple of simple thought experiments, is what would happen if you were to apply a force to this meshwork? And this is shown here via, this is gravitational field waves, but the concept's the same, is that you've got a meshwork and then you apply a signal. And here, this is perturbation, this is something impacting on it, but it could be you switch the motor proteins on at that site, or it could be whatever it is, you cause a very localized perturbation to this system. Then what you've got is you've got these switches which are able to buffer that force by unfolding, but when they buffer that force, they also change the signaling at that site. So then the whole cell can adjust. And this domain unfolding changes the coding at that site, which then can adjust the behavior of the cell and help it to maintain homeostasis. So with just these Talin and vinculin as a single mesh code, not worrying about all the others, there's incredible diversity and nuance in the complexes and the signaling you can achieve by making different arrays of these switches and different numbers at different stoichiometries. And because maybe what I also forgot to mention was that there's 11 vinculin binding sites on this molecule. So you can have between 11 vinculins coming in and decorating on each Talin. And then, so there's a huge number of arrays of stuff you can build. But two more simple questions, which I think are both true, is it's easy to imagine that every single tissue in the body has got its own mechanical properties. We can measure that, that's physically measurable, even like, it's true. But even within the cells, they've got different contractility, different mechanical properties. So it's not hard to imagine that in each of those types, you've got different mesh code patterns which conform and respond to that. And then the second thing, which is definitely true, but a what if question, and it's shown quite nicely on this side. And this is a proteomic data set. And blue means there's not much of a protein, red means there's lots of a protein. And this is different regions of the brain, but it could be different regions of the body. But different proteins in different regions, different cells in different regions express different proteins. That's well established. It's not hard to imagine that in here, there's things which either directly interact with the mesh code directly, or impact or regulate the motor proteins which pull on the mesh codes. So it's not hard to imagine that you've got these two different things altering and responding to this complex coding. So it enables this incredibly diverse and nuance in this, what's called integrin signaling. And what I would like to suggest is this isn't integrin signaling at all. The integrins are just the readout of the ECM. What we're actually looking at here is a mesh code computational layer where all of these switches and all of these positive and negative interdependencies between all these molecules is the computation which is going on. So what I've spoken about so far could easily just be general mechanotransduction, cell migration. The last nice trip I had before COVID, went to Australia in 2019 in November, sat at Sydney Airport and just had this epiphany moment of a different way of looking at this. And what we think about in terms of mechanotransduction is that we've got basically this balance of different forces between the extracellular environment and things moving, and then the cell's force generation machinery. And as these push and pull, the solution of the string of switches is the sum of both of these these contributions. But if you ask a very simple question of what would happen if the extracellular environment is constant and predictable, and it's really important to stress it, I'm not saying it's fixed and never changes, because that's definitely not true. There's huge amounts of ECM remodeling, and the cell invests a lot of its resources in getting the ECM just right. But it's all controlled by the cell itself, by the system. If the ECM is knowable and predictable, then all of a sudden we've got a very different scenario here. The extracellular forces aren't having a huge impact onto this. It's no longer a tug of war. What we've got is we've got a cell which has got a series of binary switches which it can operate with its force generation machinery. So in this scenario, we can now have a signal which switches the motors on and pulls and alters this pattern, controlled by the system itself, not with so much contribution from the outside world. And that was where the penny dropped and led me to write this over those next 6 months or something, that if this is an ordered system, then information can be written into these molecules by generating contractility within the cell. So this is where the idea that this is a memory molecule came from, because in an ordered system, these are persistent linkages which can form. And it's this notion that these are binary switches. And you switch on the motors one time and you can trigger from an 000, one domain will unfold. If you pull on that one again, you can't unfold that domain again. So then if you pull on that one, you'll go to an 011 or a 110. So you'll get a different response to the motors getting switched on, depending what state your molecule's already in. But what that leads to is the notion that information is being written into the shapes of the molecules in these adhesive linkages. and they're being written into the shapes of the molecules via changes in tension by the cell switching on and off its motor proteins. And if you do that, you can write information into this as a function of the prior events, because the pattern will change depending on what pattern it's in when you switch the motors on. And we can show that's true experimentally. At the single molecule level, we can manipulate these back and forwards. And because we know how these molecules respond to force, we can now simulate the folding and unfolding with high reproducibility, which we can reproduce experimentally. And what that enables us to do is you can physically manipulate the molecule to write information into it, pushing and pulling in defined quantized units to actually do sums and manipulations on a molecule. And it can store this information persistently on a single molecule. The key thing here is to be able to store it persistently without other factors. There's lots of other factors, but a molecule would hold its shape permanently if it was a perfect linkage, which obviously it's not. Nothing's perfect. But in terms of how the brain can function, is if the mechanical environment is well-defined and predictable, then this same thing can occur. And that leads to a prediction, again, which I think is true, is that the mechanical environment of the brain is perfect. And for a long time, mechanobiology and mechanotransduction's been a bit underplayed in the brain because it's so soft and squidgy and so hard to overcome these force thresholds, I say. But this actually makes it perfect, because what that enables is for each synapse to precisely control its own mechanical environment, isolated from the others by the soft nature of it. So this enables mechanical computation across each synapse to be perfect. That's the hypothesis, that's the theory, but I also think it's likely that's the prediction. And if we go back to looking about what these synapses look like, this is a terrible drawing. Every time I give a talk on this, I always think I need to remake that because it's horrible, but I will do it one day. But in essence, what you've got is you've got a synapse where you've got the two ends talking to each other. You have an electrical signal in one causes vesicular release across. And in that scenario, if you, this is scaffolded via these switches. So if we look at what this signaling is occurring through the middle, but all around the periphery of this, you've got this complex scaffolding built of binary switches. And a really fantastic bit of advice from one of the reviewers of the MeshCODE at a Glance paper was to actually write these as the binary strings of information to help clarify that this is binary information written into these molecules, the shapes of these molecules. And so around the edge of that synapse, you've got this binary information. Once you have a signal go through the synapse, it switches on the motor proteins, as is really well established that you get contractility following synaptic vesicular release and uptake. Then that switches on the motors and physically pulls on these adhesions, causing these switch patterns to change. As you recruit more vinculin to these vinculin's a one-state binder, it cross-links to more actin. You build these adhesions, they grow as a function of signaling, and they atrophy as you let go. And also what we tried to do was to clarify this even further and strip it down to, instead of showing all of these linkages, to just consider these Talin molecules as an array of these switches. And this is just showing 8 for simplicity. There's probably at least 100. We think there's more than that in one of these adhesions. But prior to the contraction, it's in a certain state, pattern of 0s and 1s. But once you pull on it, it then begins to start a calculation process. And this whole, these adhesions are a calculation because the whole thing has to balance out. And it has to give, I think there's solutions to an adhesion. And each time it's explicitly dependent on the Talin switch patterns as the core of this thing. So the stimulated adhesions will grow in a pretty quantized manner because of these switches. And if they're not stimulated, then over time they will atrophy, exactly as what we see in cells in culture. The adhesions which are stimulated grow and the other ones shrink. And this is what I think is actually happening in the brain. You've got this stimulated synaptic adhesions which grow. So the signal across stimulates them to contractility, pulls on these adhesions, they start to grow and mature. And that might be exactly what's happening in there, exactly the same as what we see in adhesions in culture cells, where you send out filopodia, you start to build adhesions, then you start to build more complex structures. And all of these structures are explicitly dependent on integrins. So by extension, they're explicitly dependent on Talins because you don't have integrins active without the Talin molecules. So that leads to this notion that what we're looking at, and the idea I'm trying to present here, is that this is a binary coding built into each of these synapses. And in essence, that means that the brain is a mechanical computer, is what I'm trying to argue. And the synaptic transmission going across this updates this coding in both the pre and the post-synaptic mesh codes. And this can update this as information, but it also tunes the activity of that synapse. So not the information actually encodes the activity and the likelihood that synapse will fire again. But what's exciting to me is that every cell in the body has got these same mesh code structures. Obviously, the ones in the rest of our body are not as dynamic as the ones which I would suggest are happening in the brain. But this would amount to a machine code coordinating the whole organism. This patterns of ones and O's would be helping every single cell in the body to work in synchrony and as a collective working to the same code, essentially. And we got, I've been through this already, so I'm going to skip through it. But in terms of the synapse, what you could imagine is that architecture of those discrete, isolated synapses on a stick is perfect because it means that each one can behave as its own unique signaling hub. And as you pull on them, as you stimulate the motors, you can recruit and displace molecules from one synapse, which can then float off to another. And as I've already said, that stimulated synapses would grow, unstimulated would shrink. And if you imagine these switches across the whole of the neuron, then what you've got is you've got an array of switches in that neuron, a finite number. And so you'll get redistribution of these molecules, these signaling molecules around that neuron as a function of the switch patterns, as a function of the activity of the individual synapses. So you'd have a different pattern of folded and unfolded domains in a stimulated adhesion. So the signaling would be different, exactly as what we see in cells in culture. And again, some of these drawings should never be allowed out of my house because they're horrible, but this is another one I drew in haste, which ended up becoming one I used. But this is just to try and exemplify some of these potential ideas. And this is showing 4 different synapses in 4 different states of activity. And in the top, we've got one which is just being triggered by this is supposed to show the electrochemical signaling. The hypothesis here is that the electrochemical signaling across a synapse switches on the motors. It causes this contractility in a quantized manner at the stimulated synapse, but not at the unstimulated synapse. In the context of vesicular release and on the presynaptic side, it's not hard to imagine, and this is something which we're actively working on, is that if there's components of the vesicular release or the tethers or the recruitment, which are sitting on these switches, then if one of those switches unfolds as a function of force, you could essentially like pull in the trigger and releasing it. So this might be in the presynaptic side, it could be involved in the vesicular release. And that what seems almost like random, the probability of vesicular release, it could be explicitly dependent on this mechanical computation. Something which is really well understood and really well recognized, but not well understood, let's get it the right way around, is this concept of synaptic tagging and capture, where a stimulated synapse can recruit proteins, newly translated proteins to that synapse. And it's really mapped out that this happens, but not quite sure why. In the MeshCODE theory, this becomes totally obvious because if those newly translated proteins are being recruited to switches, certain switch patterns, then when you stimulate it and you unfold a domain, you might recruit certain proteins to it. And that would be why they go to that synapse, but not to this synapse. And simultaneously, you've got this inverse synaptic tagging where you've got molecules which are recruited to synapses to keep them in a dormant state. And if they recognize the switch in the folded O state, then when you stimulate one synapse, you'll kick that dampener off and he'll go and accumulate on the unstimulated synapse. So you can be constantly shuffling these molecules around as a function of these signals. And then as a synapse matures and becomes a full-blown synapse, then further stimulation will cause contractility and it'll update these patterns further. And that's what I propose is the where memories are stored, the location of the engram. It's these physical changes to these shapes of these molecules, building these different signaling complexes at that site. So what this would mean is that every synapse would be constantly updating with new information. And this is a standard reverberating circuit, just to highlight. But if you imagine that in each synapse at the start, you've got a certain pattern of ones and O's, here I've drawn them as reds and whites, then you stimulate that circuit, then you trigger the synaptic release and you trigger this mechanical computation. And that then, part of that computation, the pushing and pulling and these motor proteins, leads to other synaptic releases, so that this only balances out when the whole circuit balances out. So this can switch on whole circuits of neurons. But then at the end, what you've got is you've got a circuit where the calculation is completed. In that state, the pattern of information is different because the synapses have been updated. And this might be that this is a learned thing, or it might be constantly updating in terms of things like dead reckoning or navigating or all these other type of things. You could constantly be updating this calculation. But in this MeshCODE theory, this information would control the synaptic strength and the probability of firing. So you could switch on and off different parts of the circuit via these same simple mechanical rules. And this leads to this notion of a way to input information into the brain, and this is well established for like, what is it, like 60, maybe 100 years, is that these action potential spike trains, these electrical signals which travel down the neurons, are a way that the sensory organs send information into the brain. So this guy is just getting into the shower. He wants to know whether that's too cold or too hot. He dips his hand in tentatively. The nerve endings in the fingers detect that, and they send nervous action potential spike trains to the brain. It calculates, if it's really, really hot, his hand will come out reflexively, but otherwise he'll make a decision, oh, it'll be all right. But at each stage, the calculation occurs here, but at each stage, the information will be updated in all of these synapses on the route. So the animal will be constantly updating this binary, what's in essence is a binary coding. And this is just a guy getting into the shower. But what you could imagine is you could imagine that you could have an entire mathematical representation of an animal's unique life in this coding in exactly the same way. From the second that the baby is in the mum's womb and becomes viable and the brain's mostly formed, it can start to write information into that. So you could have a generic default state with all of the like standard operating system, so to speak. And then from that moment, depending on that individual's unique life, it writes in its environment, its interactions, what it eats, what all of this stuff to specialize that animal in the context of its environment and its unique life, all written by updating these shapes and these pathways of information through the brain. So I'm on the home straight now, you'll be pleased to know. So I'm going to wrap up, but just a few thoughts, which I think are quite important. And they're mainly just thought experiments. But if you could imagine that mechanical switches can each can store one bit of information, then if you build an array of switches, then you can store a vast amount of information, exactly the same as what we do in computers and in silico. And what's really striking, and this, when I looked at this, it made my jaw drop, is if you look at the structure of the brain and look at the structure of the cortex, this is I love this drawing, it's from 1996, but I think it's absolutely gorgeous, is that the brain, the cortex, is incredibly logical. It's got 2 million of these cortical columns, repeating units of highly similar architectures. And each one of these layers is precisely defined. And each of these teardrops has got up to 60,000 different synapses. Each of those synapses has got 100 different talins. So there's a vast array of switches, mechanical switches, arranged in a highly logical way. And if you look at how our best efforts at high density data storage and the amazing advances in solid state devices, then the similarities in the logical architecture didn't escape me. They're sort of mind-blowingly similar, in that we've realized that the most logical way to write data is to have an incredibly logical, repetitive structure, exactly like in the brain, in essence, where you've got many, many channels. Each of them's got many of these NAND flash, which have got pages and blocks and then switches. So in this solid state disk, there's a memory controller down here, which writes information into these specific regions we're giving it a specific address so that it can be found. What I'm proposing here is exactly the same is happening in the brain, that each of these synapses can be specifically addressed and information can be written into them in a similar fashion. And I had to get my son to help me, but this is just as a back of the envelope calculation. If you just worked on 100 Talins per synapse and just said 8 active switches, then you could have 10 petabytes. That's the back of the envelope. I wouldn't put too much weight on that, but it's a vast amount of information. And one of the problems, I don't know if you've had a computer for more than a few years, if you want to look for information from a few years ago, it's really hard to find it unless you've got ordered logical file names. So if you do have petabytes of physical data written into a disk, then there's got to be some way of allocating that and mapping it and giving it an address so that you can keep it stored, but only call it when you need it. And that's a challenge for any sort of discussion of how neuroscience might function, is how do you address and map the information in a way it can be allocated? Because you get some random information which you store in the dark recesses of your head until you go to a pub quiz. They ask for this random fact and all of a sudden, bing, you know it straight away. But it's not looping around your head the whole time. And that's because it's addressable in memory, is what I'm arguing here. But if that's the case, then you need to have some form of file allocation table. You've got to have somewhere where there's a map showing which bits have been wrapped to where in the brain. And what's emerging from studies, not from me, but from loads of labs all around the world, is that the hippocampus is looking more and more like the immediate day-to-day computation and the memory controlling unit, really. Because what's looking at what happens is that you get information in the day, it's temporal in the hippocampus, and then overnight it gets written into the cortex. In the MeshCODE idea, that writing from the hippocampus at night, it would suggest that sleep is a key part of the MeshCODE. It's involved in the data management. And what happens is when you first go to sleep, you go into a very deep sleep, and that's to now enable these mechanical calculations to finish in this theory. And that enables them to complete and get to that homeostasis, mechanical homeostasis. But then you go into a deep sleep, and then all of a sudden you have a lot of electrical activity. And in this REM sleep, in this model, this would be the brain physically writing to the cortex. So you've got a lot of this electrical activity, and it would be switching on the motors in a highly ordered way to read and write and update these patterns in the shapes of these molecules in the cortex. And then once that's done, you go back into a deep sleep again to allow that calculation to finish. And then you can do more data management over time. And that might be why sleep's essential, because the sleep overnight, the memory traces are consolidated and these switches are updated such that the synapse, the flow of synaptic electrochemical signaling will be tuned based on this. And it might be why if you do an all-nighter, the next day you are very not so good, because you've got fragmented information. It's not written it's not consolidated. So that might be why sleep is essential in this theory. So to summarize and to ground it back, because that's the extrapolation of the theory, but to ground it back on the experimental work, we can show that Talins have got this molecular memory. We can show that they can store data persistently in a binary string, and we can show that that switches on and off different cellular processes as a function of those switch patterns. What's exciting to me is that this is all based on really well-established biophysical principles. It doesn't require any ethereal stuff or any delving into the quantum world. This is biophysical principles. But what it also means is it identifies the physical location for data storage in animals written into the shapes of these molecules and the complexes which assemble on top of them. And as I mentioned before, it also leads to this notion that this MeshCODE might be a machine code that is keeping all of our cells running in synchrony. The brain might be very dynamic, but it might be sending out longer term mechanical signals to the rest of the body, keeping us all working as an organism. What I think is really the best thing about this is that this is completely consistent with everything which everyone's done on electrical and chemical signaling. The electrical chemical signaling is well mapped out, but in this model, the information processing and storage would actually be this mechanical layer under the hood, where the electrical and chemical signaling is updating these switches and updating these patterns as a result of this mechanical computation. So what I think is that this identifies a binary code and an addressable read-write memory system. And I think that that supports the view of the mind as an information processing system and an organic supercomputer. Or an organic mechanical computer cluster maybe, but a really complex computational device. And I will thank you for listening. I think I might have gone over slightly. Sorry for that. But just a massive thanks again to Ji and Ming Chi. This has been the best journey ever because every single experiment we've done has just opened up a whole new landscape of what's going on. I'd also like to thank Martin Schwartz and Nick Brown because they've been pretty critical of this area and a lot of the times and helped ground it and helped to develop it. All of the guys in the lab are helping us to prove it. And my old mentor, David Critchley, who's still super helpful all the time and offers good, nice feedback. And I'll just finish with a blatant sale plug. If anyone wants to come and work on Talin memory molecules in the lab, then we've got a 4-year postdoc position, which is just about to be advertised this week. And it's part of a CRUK, Cancer Research UK programme grant with Martin Humphreys in Manchester and Klaus Jorgensen, whose name I've spelt wrong, at the CRUK Institute in Manchester. And anyone who's interested, please feel free to email and the advert will go live soon. Thank you very much for listening.
Thanks very much, Ben. That was a really interesting talk. So the floor is open for questions. There are a couple of questions actually in the chat.
I just want to make a quick comment for the QLS students. Please don't run away. Remember, you're going to stay and chat a little bit longer as well with Dr. Goult. But yes, go ahead, Anmar.
Okay. Celia, do I read the questions or shall I ask the people to actually ask the questions directly?
Benjamin, do you want to ask your question?
But there's one before Benjamin. There's Julian Willett.
Yeah.
Julian, do you want to go first?
Yeah, are you able to hear me?
Yes.
Oh, okay, yeah, so thanks for the talk. It was very, very interesting. I just had a question from earlier in the presentation and when you were discussing the different conformations of the Talin molecules. I guess just from like a physical chemistry / biochemistry perspective, you often touched on how there were 2 conformations, but were there just 2 conformations that were most stable, or were there a larger number of conformations and those 2 were just the ones that had the lowest free energy? I was just hoping to better understand that.
Yes, that's an excellent question. And there has been some, so certainly in the single molecule with the resolution we can get, we see that it's a sharp transition from a folded to an unfolded. There's one of the domains R3 which fluctuates, but most of them are either in that state or that state. Some of the computational modeling and the molecular dynamics have identified a potential 3rd state of a 3 helix bundle, but we don't see that experimentally, but there may be more, maybe the 2 major states. And then there's arguably another state where if you pull really, really hard, then the helices becomes a proper full spaghetti. But then the whole thing would disassemble then anyhow. So there's 2 major states, but I wouldn't rule out that there's others as well. And also on that note, I would say that one of the things which is interesting to us is that there's post-translational modification sites on some of these sites, and they fix patterns and they stop certain domains refolding. And it might be that if vinculin pulls 1 helix out, one of the things we're interested in, maybe they could fold into a different domain with we don't know the full extent of it yet. But I wouldn't rule out there being intermediate states. But yeah, it's a good question.
Okay, Ben, you want to ask your question?
Yeah, can you hear me?
Yes.
Yeah. Okay, so thank you very much for your presentation. I have a question about the MeshCODE model. So I'm not sure if you mentioned this in the presentation, but you mentioned it in the paper that each Talin protein would represent a bit of information. I was wondering if with the dynamic nature of the actin network, could the meaning of each bit change due to network remodeling? And if so, how do you think cells would account for that?
Yes, so a bit is assuming, that was assuming, a byte was assuming that 8 switches were operational at any time. I think the actin dynamics is but the actin dynamics is directly coordinated by the Talin switches anyhow, because the more vinculin you recruit, the more actin linkages there are. So the dynamics will be taken into account. But I also think that these systems are much more ordered than what we see experimentally in culture. because in culture, these cells never reach their end state fully. Even when you build a synapse we're doing a lot of work trying to build synapses in microfluidic chambers, they never reach the fully finished state because they never have all of the perfect ECM they need. It's never quite perfect. So I think there's a slightly, I think that the dynamics of the actin cytoskeleton, I reckon that it's more stable than what we think, but that will definitely have to be factored into this for sure.
Okay, there's a question from Eric Cook. Okay, Eric Cook, yes, please.
Interesting idea.
You muted yourself, man.
Interesting ideas. I like it. So maybe you mentioned this, but maybe I didn't catch it. So I was to say, has the link been shown that if you take a synapse and you change these switches, it does change the synaptic strength or the basic, pathways from release to postsynaptic response in some ways? I mean, I know you showed like these molecules are part of the structure of the pre and postsynaptic structures as you're shown here in the diagram.
Yes.
Has it been shown? And if it hasn't, any ideas how you would do that experiment? Like, how could you go in there experimentally and flip a bunch of switches and then show that now the synapse is functioning different?
Yes, it's definitely a challenging experiment. So we're trying to do it with microfluidic chambers where we can stimulate a synapse in the control and then try and look for changes on the postsynaptic side with a fluorescent vinculin molecule or with to try and show changes, or with the actin cytoskeleton, whether it grows or whether there's tension on Talin. We've got some drugs which target Talin, which we're hoping might give a lead on it, but it's definitely a challenging problem. But the other one might be to watch the actin cytoskeleton grow, because it swells out as you stimulate these things, and that's dependent on that's an adhesion dependent effect. So to actually prove categorically that it's a switch change, that's the goal. Yeah, that's exactly what we want to do, and that's what me and Emma have a grant earlier in the year to try and prove that. It's the big challenge to actually show that what happens in a cell in culture bears out in this, and that the memory molecules are altering the activity. But the other thing which we're doing, which will help with that a lot, is I don't want to go into too much detail now, but a lot of the Talin proteomics we did from brain identified some quite promising candidates of presynaptic molecules. Let's leave it at that. So we're hoping that if we could prove that there's a molecule which binds to a switch, we can mutate that switch and then we no longer see activity or we see altered, then that would be a step towards it. But it's definitely a challenge for sure.
Okay, thanks. No, that's good.
And one other thing as well, one of the things which might be fun to try, which we're hoping, which we don't know if it'll work yet, is that if we can detect this mechanical fingerprint, then we might be able to improve on the probability of physically released. We might be able to preempt which one's going to fire next, because we might see that there might be a fingerprint on the Talin. So we're looking at a lot of the tension on Talin to try and tease that out. So yeah, it's a work in progress, but it's, yeah, good question.
Okay, there's a question from the room. Just go ahead, Alexander. Can you give the...
It's Lucas is going to ask the question.
Hi, thanks for your presentation. I wanted to ask, so you talked about the different R domains. Are the contour length increments for each R domain unfolding the same? My question is about like tension propagation theory in the context of pulling a protein that has many units, many folding domains. To me, it would be very hard to unfold an internal R domain, for example, R7, without unfolding R1 through 6. Because if you pull from the R1 side, the tension is going to be highest in R1, slightly lower in R2, slightly lower in R3, et cetera, as you move towards the other side of the protein. So, you showed a lot of diagrams of strings with zeros and ones. In my mind, it seems like you'd have blocks of ones followed by blocks of zeros, and depending on the tension, the number of digits in each block would change. I'm wondering if you could comment on that. And if you're in your experiment, are you able to determine which R domain is unfolding from the force extension curve data?
Yeah, so it's a good question. I would challenge the fact that you would have tension from the R1 and going down like you argued there, because if you hold a molecule under tension, if I was holding a piece of string with someone in the lab or over there, and we were both holding it under tension, then if we pull, the tension's across the whole of the thing. And each of these switches have got different mechanical responses. So there's some which are really easy to open, some which are medium to open, and some which are really, really hard to open. So the order, when we first did that experiment, I thought we'd be able to say exactly which one was which. But now we can say it's going to be one of these, and then it'll be one of these, and then it'll be one of these. So yeah, they do go in a specific order, and we can, God, there's a lot of stuff. So I'm nearly there. I should have just exited out and found it. Yeah, so we could say that this, so we ended up binning them into different categories: the very, very weak, the medium, the strong. And incidentally, the observant ones, this double step here, this is the R7, R8, which is essentially got a domain inserted in it. So when it unfolds, you see a big jump because it's basically two domains unfolding at once. But yeah, they're in a different order. It's not one, then two, then three, then four for sure. No, it's not. I don't know if that answers the question, but it's more complicated than that. And it also depends on the ligands which are bound to it, because ligands which sit on the outside stabilize it. The vinculin which binds on the inside in the open state limits it refolding. And also it gets more complicated again, because when you activate actin binding here, then the forces are different. The forces act on this bit and they act on this bit like differently. So the force, where the forces is going is not purely linearly, but at the end of the day, it's still, we end up with a certain pattern of folded and unfolded domains. So trying to tease out that, and one of the things which we're trying to do to prove that in a minute is to pull orthogonally on this and see, because that you'd imagine there, then the switches nearby would feel that much more dramatically. But pulling end to end, I don't think it's the case that the force will all be at one end, because the whole molecule will be under tension.
Okay, thank you.
Thanks.
Any other questions? I'd like to ask a question myself, actually. So when you apply the force, actually, let me motivate my question first. You mentioned the word hysteresis a few times.
Yes.
So did you actually detect the hysteresis directly by looking at a decrease in force rather than an increase in force, and see if you observe the hysteresis?
Yes. So if you pull them, say that switch which unfolds, these switches here which fold at 10 piconewtons, if you pull it so that switch opens and then you lower the force to 9 piconewtons, it won't refold. It won't refold until you come back down to like 2 or 3 piconewtons.
Okay, so it's very fine. So okay.
It's much higher. The unfolding force is much higher than the refolding force. So there's the persistence because of that. So if you hold it in a perfect linkage, it would stay in that pattern forever. Of course, that's not possible because there's thermal noise and all sorts of stuff. Yes. Where these patterns get reinforced is because the vinculin binding to 9 of these different domains on the inside, it fixes that, because once the vinculin's bound, that switch can't refold because there's a vinculin in the way on one of the helices. So these patterns build, and because they're directly interdependent on, I'm just getting attacked by a fly. So they build and they're explicitly dependent on that. So the hysteresis is really important. I think the hysteresis gives it a temporal thing of a few minutes. When we hold it, we can hold it for a few minutes. But then what you rely on is other things coming on in that time and binding or being displaced away to change the actual signaling and change the composition of that adhesion. Mm-hmm. So you don't need these molecules to be in a perfect mechanical tether for more than a few minutes, because the things which build on top of it will stabilize those patterns.
I see.
I don't know if that answers again.
And, but I'm just wondering if it's also possible then in this case to have a multi-stable scenario where for a specific force you could have multiple configurations that could coexist.
Yes, definitely. But the way that gets interesting is that there's positive and negative regulators of contractility on these, like the DLC1 and the TAM. They're opposing functions. So across the whole adhesion, this will balance out, because if you have too many of the positive regulators of contractility, so the whole thing, I think there's a solution where all of the Talin molecules are in a certain pattern, which is where the information resides, which means that they're not explicitly dependent on every single, because another thing which one of the reviewers was very forceful on was that relying on an individual switch to be perfect. But if you've got a system level, a series of switches all built into a specific pattern based on the pattern of switches, then you can tolerate things. If there's an imperfection, it will self-correct. It will be because of the feedback loops, the positive feedback loops and the negative feedback loops on these different switches is the idea.
I see. Okay. All right, thank you. Any other questions?
Yeah, perhaps given the time.
Yeah, I think maybe we should wrap it up. Okay, so I'd like to thank again Ben for giving a very interesting talk. It was pretty exciting. And thanks everyone for attending, and we look forward to seeing you soon again.
Thank you so much.
Thank you,
and students, remember to stay around a little longer, okay?
All right. Bye. Bye, Ben.